Key.
Thank you very much. I too appreciate the opportunity to share our research. I've entitled my talk, Newly Introduced and Re-emerging Pathogens, Discoveries and Challenges for Their Control. As Roxana mentioned, you know, it's constantly a new challenge, it's a dirty job, but I figure somebody's got to do it, so it might as well be us. Today we're going to actually focus on anthuriums and then also give kind of an update with syngonium and rapidohea death. Okay, bacterial blight of anthuriums, this anthracnose or black nose disease, and rod and syngonium. Okay, I too want to say that, you know, it's the teamwork and collaboration that makes all of this possible. I'm really thankful for everyone we work with the growers themselves, doing the work there, the material we can use. And I also want to really say that my lab team, their pictures are on the top. I'm so thankful that we all work together. The outline today, so I'm going to start with bacterial blight. Then I'll move on to the fungus that causes black nose disease, and then again with syngonium and an update on rod. And there's our disclaimer, yeah, just because we say specific names. We're all familiar with bacterial blight. That's actually what the bacterium looks like in a pure culture growing on a plate. You've seen that already. Xanthomonas, Exonopodus, Diffenbachia, we'll refer to XAD. And these are the typical symptoms. Usually those V-shaped lesions are coming in through the hydathodes. You start with this water soaking, necrosis, the chlorosis or yellowing. It can start very simply. It turns systemic, as Tracy mentioned, you know, going in one place and actually traveling throughout the plant and finding it in another. As already mentioned, you know, the great work done by the University of Hawaii, years and years of research to try to really control bacterial blight. You've heard, again, Roxana saying you start clean and try to stay clean with just clean material, plant material. There's a number of hopefully preventative or cultural practices that can aid in keeping the disease pressure down. They're listed there. And it's critical, really, if there is disease material, proper sanitation. But all that being said, blight's still a major problem. And, you know, you've heard about our pipeline. I mean, that's one important aspect. More on the long term. But what we were really trying to figure out, as far as with management, looking at the disinfectants, you know, we were running into an issue that the main one was no longer available. We kind of had the industry standard. Sometimes there are new things to look at. Other times, maybe there are just household disinfectants. I mean, we really had no idea how effective some of these could be compared to the industry standard. So we went through this more of a shorter term disinfectant efficacy study. And I'm going to show a lot of pictures and kind of walk you through how we were looking at what we were looking at, kind of as a three-prong approach. First thing really was in the laboratory, if you take your product and you take the bacteria and you put them together, what happens, okay? Then could we start in the laboratory actually looking at ultimately what is done in the field, okay? You're disinfesting your tools, so how can we kind of mock that in the laboratory? And then actually go to, so we had a long list, try to limit it, see what looks effective in the laboratory, how does that look in the field as far as a field management. Okay, so these were the bactericides that we looked at with the active ingredient, and again, X-Spore was becoming unavailable. LD, the industry standard, DCD, and then we started looking, you know, there's just some common household things people like to use, maybe some a little more environmentally friendly, alcohol, water as just our control, and also the idea, could we use Agristrep? And a lot of times there is antibiotic resistance found in bacterial populations, so we wanted to see how much of that was with XAD. Okay, so for the first stage, the spot plates, really, you're growing up your bacteria, you're taking the plate, you're putting the bacteria, really you're just going to grow it in a lawn. But what you do is you spot your chemicals onto it, and now you're hoping you're going to see this zone of inhibition, or the bacteria is not going to grow in the presence of these chemicals, okay? And you're really interested in determining, well, are they bactericidal, meaning they're killing your bacteria, or are they bacteriostatic, okay, meaning they're kind of inhibiting growth, but when there's no longer that chemical, the bacteria start to grow again, okay? And that's very important. Are you looking for 100% management? I guess that's something else to think about. How much of the disease is an acceptable amount? And that could also influence your management, okay? So you set this up, you let it grow. We would look at it because the bacteria are growing right away, but then after the 96-hour period, is it cidal or is it static, okay? And then actually determine this percentage of regrowth for the inhibition zone. So just to, again, I have lots of pictures, but I summarized in the table, so you don't have to catch everything, but it just kind of shows you a difference. Like right away, you can tell pine sol, there's a full lawn of growth. That's XAD growing, on the plates. And in other cases, you do see these zones of clearing, indicating, okay, there's some potential. Let's keep looking at it. As time goes on, now then this was what was tricky. Was there still that clear zone, or do you start to see colonies forming in that inhibition area, okay? So again, it's a cidal effect, it's killing on contact, whatever you're dealing with, versus, well, you're reducing growth, but you're still not killing all of the population, okay? So the idea was to really determine, okay, from a larger list and what chemicals are used by the industry, is there anything else out there? And so for an overall inhibition zone at 24 hours, was there, yes or no? And then when you took it to 96 hours, is there regrowth in that area? And so as you can see, it definitely varied, like 35% IPA, right? There's 100% regrowth. I mean, there's 100. So now you can start, okay, they're not going to be effective as some kind of management. But at this level, less than 10%, 5%, that's a good thing to carry on to the next step, okay? So we, for cut flower production in particular, the idea is to getting to disinfesting your tool. So as the first mock, using very sharp scalpels, dipping it into the bacterium for a certain amount of time, then dipping it into your disinfectant for either no time at all or 1, 5, and 10 seconds. And we figure if you have to do more than 10 seconds, I mean, it's not going to be practical, right, with your tools out there. So with that, and then we actually stab the auger 4 times, and that shows you the setup of the plate. Okay, again, this is the bacteria growing on the plate after you stabbed it, right? To see, are you getting actually bacteria to grow after being in the disinfectant? 0, 1 second, 5 seconds, 10 seconds. Okay, obviously water control, everything's going to grow, okay? But for 7 days, you can now start to see, again, the top right quadrant, it's not in the disinfectant, and then all of a sudden, 1, 5, and 10 seconds. And again, you're seeing things that are not effective at all, and you're starting to see things that, wow, certain ones are definitely holding up, okay? That was, a shorter list, and it's kind of, again, the industry standards and alcohol and Lysol are actually starting to show some promise. Using a very sharp tool, but what happened, so our thinking was, well, if we switch to a toothpick to kind of mimic the old or corroded, slightly corroded, maybe pitted, where the bacteria can kind of, right, does the chemical get to those spaces, and what does that mean? Okay, so it's the same setup, dip it in the bacteria, dip it in the disinfectant, do 4 stabs, and see over time what happens, okay? And I kind of put it to a black and white theme to make it a little more easier to see the differences. Okay, sterile distilled water, here you can see the 4 dots in all 4 quadrants, but then I've marked again where some of these are actually you're not seeing. Okay, so again, holding up with 3 days, and also then looking again at 7 days past that poke. Okay, and the summary. Okay, so after a certain amount of time, I've kind of highlighted now the chemicals that are working really well. The 0%, so we're actually getting no growth of bacteria, even with the toothpicks at certain times. But as you can see, this pitted nature, or the areas that the bacteria can hide, may be a little more difficult to get the disinfectant in. Sometimes then you would have 1 or 2 starting to grow. Okay. And then finally, it took us, knowing what we did, we could limit what we were going to look at with this type of method, actually setting up the same way, but using the shears, put it into bacteria, dip it into the shears for a certain amount of time, and cut twice on leaves, okay? And then repeat that process. And we also did then 3 leaves per plant, and we also randomized the age of the leaf, okay? So we wanted to try to catch everything. We looked at Midori and Marion Seaforth, kind of our favorites from our pipeline. And I'm just going to quickly go through some of these pictures. After 35 days, okay, these are when all of these pictures kind of show, you see your controls, and again, those V-shaped lesions, the water soaking, you see necrosis, chlorosis, okay? And then with a 5-second dip, this Marion Seaforth and LD, it actually starts to limit or eliminate the bacteria from causing a problem. Okay, this is Midori and LD, Expor and Marion Seaforth, and so maybe there's just very, very mild in one case, that upper right. Okay, again, in one instance, but in all of what I'm showing you compared to the controls, I mean, there's a huge difference with these. So LD, DCD. Okay, the alcohol, 70%, 91%, very effective. Okay, again, left side is the controls, right side's Midori with the alcohol. Okay, and surprisingly or not, Lysol actually held up really well too. That's Marion Seaforth, this is Midori. And this is the results, okay? So we're actually just going to follow up with one more experiment to see, well, how long does that solution, once you follow label rate and now you're using it for your tools, how long does that remain effective, okay? So we definitely have some options. I mean, what is being used and potentially what could be used, okay? Really, again, so for bacterial blight, prevention rather than a cure. That's the case in most diseases. You're trying to delay the onset of disease and actually reduce that initial. Start clean if you can. Also then reduce the rate of disease development. I mean, if you're knocking off the population by 95%, that definitely has some potential, okay? So while some of our other projects really are these long-term, this was a chance to really see a short-term type of experiment and maybe potential of some new options, okay? So the title, with either emerging or re-emerging, this is, as a pathologist, what seems to be more common these days. I'm going to talk about black nose disease of Anthurium. Okay, again, I want to say thank you to, but I was thinking, it's like thank you, Brian and Joanne, for pulling us into, and I was like, wait a second. No, it really is this great collaboration of finding things that we can investigate a bit further, yeah. Anthurium black nose disease, the history, it's caused by Colletotrichum gloeosporioides, okay? But this list, what we're showing is now we found more species of Colletotrichum and even a Phomopsis species that it looks like the disease, the symptomology is the same, and actually it's something else, okay? It's another species, it's another genera, and some of it was really alerted because management, it was not as effective, okay? So we were trying to figure out, are we looking at the same thing, and are the fungicides being used effective in the same way against all of these different isolates? So this is really preliminary. If you see, when we started, not even 1 year ago, collecting these isolates from different varieties, doing some of the molecular, the morphological, trying to figure out, I will say the disclaimer, you know, what has happened with molecular, it's gotten kind of tricky with these genera of fungi. So Colletotrichum, gloeosporioides, species complex. Well, it's like, well, gloeosporioides is a species. Now you're saying it's a species complex. So we're really trying to figure out how closely related these isolates are. But we found Theobromycola. Then we found those gloeosporioides. Then we actually found a fruticola, so 3 different species we think of Colletotrichum, and actually Phomopsis, a whole different fungal genera causing the same looking symptom. Okay. Originally, and then the comparison of what these other species look like. So actually, when you start to purify them and grow them on the plates, is what we're looking at here, you start to, well, okay, they're not the same. So that makes sense. And these pictures are the above. You're looking at this is the coloring underneath. Some of it's due to the spores being produced at what time. And this is actually, I mean, it looks completely different, Phomopsis. I just thought that was cool. I mean, for Roxana's cool video, right? Like, wow, when you actually look at these fungal fruiting bodies and this spore, it's not a good thing, but the pathologist kind of thinks it's cool. And we're kind of midway trying to prove pathogenicity. Okay, so that's key. Just because you find things doesn't mean it's actually the pathogen, right, causing the disease problem. But we do know at least one of these other species does the same thing. But just to kind of walk you through what these samples were that we were receiving then, and isolating and purifying to get to these multiple species of Colletotrichum. Antoyama peach, Kozahara, okay, so different colors, didn't matter. Again, the symptomology looks really similar. UH2271, a closer up picture just of black nose disease. Okay, there's actually, off of Midori, there was a speck that when you look more closely, you can actually see these fungal structures called setae with a certain color spore, okay? And so you can see there's black nose symptoms, you know, there's one and these, and they look the same, but this is one fungus, and this is actually a whole different, so this is Phomopsis. You can tell it looks really different than Colletotrichum when you start looking. That's kind of these tendrils of spores, okay, reproducing differently off of Midori. So now we knew we were working with these isolates, and we also then really quickly wanted to see if the fungicides being used, and we're thankful Brian had a lot and helped set up these plates that we could then test in the laboratory. So what this table is, again, it's preliminary, but it does show some important information, okay? The 2 different isolates of the same Colletotrichum. Okay, so in this case, there are 2 Theobroma isolates from just 2 different samples, okay? Head to head, so they're supposed to be the same fungus, right? And when you see the control, so this is just growing on a plate, and after 7 days, how big does that culture grow to? Okay, almost 70 millimeters. Now you can compare when you've amended your media, how does that inhibit growth? Well, of course, you're hoping to see, oh, it doesn't grow at all. But In some cases, there's definitely a limit of growth, okay? But what was striking right away is that the isolate 1, yes, was inhibited greatly by these chemicals. Isolate 2 essentially wasn't being touched by those fungicides, okay? Which, yeah, there was an issue. Okay, using something like that on that isolate is going to be, I mean, not do anything, actually. Then you can see where there's similarities in the chemical classes, and then these, you know, yes, there is a reduction, and they're behaving similarly. So that really, with the message, isolate variation. So we thought it was black nose disease caused by Colletotrichum gloeosporioides. Okay, the symptomology is the same. It's a huge problem for cut flower and potted flower. Suddenly we're isolating other species, a whole new genera. Okay, now we're wondering, well, is this now a new complex, right? Black nose complex. So that's work in progress. Okay, and so I'd like to think that, well, these are positive, right? I mean, even Roxana showing positive results, even though the subject matter, and try to keep it more positive in the doom and gloom of rapid ohia death, but really, in a way, you know, speaking positively for the nursery industry. Just the background, everybody knows how important ohia is, yeah. Irrigation, drinking water, culturally, I mean, high significance, and a lot of variability, which is good, and why we're seeing not the entire forest dying in locations. Just to show you the map where ohia is in certain areas, yes, it's been devastating. Okay, this is kind of the summary of rapid ohia death. So there's widespread mortality on Hawaii Island. It's characterized by this rapid canopy death. The leaves brown, remain attached. This kind of goes after, so from the healthy recent, so this is this wilt that occurs, and then say within a year or two, the leaves do fall off, and then the fine branches after a few years. So when you suddenly come upon this, it's really hard to diagnose. If you catch it where, wow, the symptomology goes from healthy to dead, definitely points to Ceratocystis. In particular, if you could take a look inside and you see this internal discoloration of the wood actually caused by Ceratocystis. So rapid ohia deaths, kind of this umbrella catch-all of 2 new wound colonizing Ceratocystis causing death in ohia. Okay. Ceratocystis fimbriata, I already mentioned this species complex. Here's another one. Oh, it's fimbriata species complex. Wide distribution, a lot of agriculture and plantation forestry. Okay, it affects many hosts. It needs a wound to cause disease. It goes through, there's a number of symptoms, important treated diseases that are not found here. Okay. But there had been Ceratocystis fimbriata already present on Hawaii Island. And this sometimes gets confusing for people. Well, it's Ceratocystis fimbriata, that's Ceratocystis fimbriata, that's the pathogen. Okay, no. And we set out to prove the no as well. It was already here as black rot, a sweet potato, it continues to be. Okay, Syngonium. So actually in the late 70s, Syngonium was found as this mild pathogen on Syngonium. Genetically speaking, elsewhere in the world, it is related to the pathogens causing major diseases elsewhere. Again, is it the same thing? Well, no. It's called the same thing sometimes, but it's not. But that's the closest related isolate, okay? Then there was something off of taro, and now you had these 2 ohia. Okay, what these 2 species actually look like growing on a plate, just like those other cool pictures to me. More fungal spores, you know, how they're born, what that means, how long lived, you're all thinking management. And then the typical pathology, just showing exactly in the greenhouse or growth chamber what's happening in the forest. Okay. The two species, when you come across a dead canopy, it's hard to tell, but when you start to get inside, for the first species that we found, we just called it A. There's characteristic staining. Typical, if you cross-section it, it's a starburst pattern. It's much easier to identify just based on visual observations. Okay. The other one is kind of this minor, elsewhere in the world, biology is not well understood. However, it is killing our tree. The staining is not as obvious. The patterns vary a lot. This is actually on the left side, widespread mortality and much more aggressive than what we find as the species on the right. Okay? Doing a lot of work trying to figure out, well, does it affect other things? Usually it's a wide host range. What else is it going to kill? So other ohia, other polymorpha, other metrosideros, but because it is a huge agriculture and nursery problem, what does it do to things that we have growing here, okay? Syngonium. Syngonium was kind of tagged early that, oh, it was a syngonium pathogen that now is killing ohia, okay? So we set out to, well, figure out if that was right. Was syngonium actually acting as this carrier host, that it came in, it's existing, I mean, it was found, identified late 70s. We went looking again and found isolates and started looking and doing comparisons. And if we inoculate syngonium with the ohia pathogen, what does it do? And if we inoculate ohia with the syngonium pathogen, what does it do? Okay. Right away, I was hopeful. They really looked kind of different, maybe to the eye when you start staring. I don't know if that was wishful thinking or not, but inoculate into ohia these two isolates, and right away we could tell, you know, something, the ohia isolate on ohia started to spread. The syngonium isolate, there's just, yeah, I mean, there's that wound you put into it. And this is what we ended up seeing. The ohia isolate on ohia, well, it kills ohia. Okay, and it that internal discoloration. It produces these spores that are actually long-lived, hard to kill, persist in wood a very long time. But the the syngonium isolate, which was different genetically when you actually start looking at it, actually did nothing, okay, to ohia. That's kind of the, when we reverse it and put those two isolates onto syngonium, well, the syngonium isolate could then easily take out syngonium. You're inoculating at a high concentration, but the ohia isolate actually didn't do anything. And we attribute to the one little dead leaf that we did see more of a senescent thing due to just the wounding, but no spread, no outward symptoms, no spores found inside, okay? I'm finishing up, I promise. Really, just to reiterate, rapid ohia death is now actually two diseases, okay, called ohia wilt and ohia canker. One is much more aggressive than the other. That's our species A. They have, I mean, they're found throughout Hawaii Island. Okay, that's the breakdown of the two species and where they're found. I'm sure most of you have heard the bad news. It's now in Kohala. Okay, that's the visual symptoms outwardly are similar. Inside, they do look different. But Ceratocystis that was here from syngonium and even the taro and sweet potato do not cause rapid ohia death, okay? It's an invasive pathogen. We're still trying to figure out where it came from and what it came in for, to stop that process. And we're also trying to figure out, well, what other things are vulnerable to attack and what can act as these carrier hosts, okay? So we'll stop it there and say thank you very much. And if you have any questions, I know we're like, but thank you.
Does the Puccini rust, which affects Myrtaceae and eucalyptus, does that have any effect on rapid ohia death?
No, so that's a really good question. The rust, it's funny, guava rust, ohia rust, actually in the wider world realm, myrtle rust, it actually doesn't do anything. There is no association of the two pathogens. They're completely separate. No, so actually the rust, Well, the rust, the seedlings are very susceptible, and the very young leaves of mature trees can become, you can see the symptoms, the rust pustules on mature trees, but that's completely different and not connected to the Ceratocystis fungi that are killing the Rapidohea death.
And that's what Puccinia is the one, it's caused the death of a lot of the rose apple trees?
Yes, yeah, so that rust fungus has a much wider host range than what we can find for the Ceratocystis currently.
Do you have any, the Department of Ag is supposed to put an embargo on the Myrtaceae. Have you heard any progress on that?
Not specifically. I know it is in the works. That's really, I'm not sure the time frame, but you're right. As far as importing, right now you can import Myrtaceae, but that's supposed to change.
Thank you.
Johnny, didn't they just vote on that at the Board of Ag meeting last week?
I didn't see it in the agenda.
I was just curious, could it have evolved? Like, could it have been something else they got here and then evolved to something to attack the living?
Okay, so could it have evolved from something here? No. And the reason I say that is because we have a huge isolate population that we've done genetic, the testing on, and they're clones of each other, which actually means, so they're genetically identical. It's the one came in and has spread, and that is known for exotic or invasives. It hasn't been here long enough to actually show genetic changes. Yeah.
So, for my ohias, in my backyard, not the forest.
Yes.
JB's already told me 100 times I can't do anything about the forest. Can I do anything about my backyard ohias?
Your healthy backyard ohia?
Yes. Can I keep them healthy?
Really try not to wound them in any way. There's a clear correlation between any kind of wounding event, including animal damage, including weed whacking, landscaping, pruning that's not done well, and having a higher likelihood of becoming infected. And that's as long as the fungus is actually present in your area. So it's kind of, the fungus has to be there in order to cause infection, but it also has to get in to cause disease, if that makes sense. So there are things, yes, as a homeowner, you can avoid your pet pig from scraping around, and you can not weed whack, have your border that you don't come near it. And you can't account for Mother Nature, but that's the...
Another question, treatments. Is there anybody investigating? I'm sure there is.
Treatments, the Rapidohea death, yes. So we are always testing and on the lookout for anything. Unfortunately, these diseases around the world, there's no cure. So once these Ceratocystis get in, again, it's that prevention, how do you, so there's kind of that two-pronged. Well, are there things, just because there is no cure doesn't mean we're not looking for one, okay, I should say that. We're investigating that avenue, and we're also investigating, because there is a lot of infected wood, a lot of infected sawdust, how can you actually sanitize that material? The same, like high temperatures or with steam, or what's the potential to actually kill everything in wood to make it usable and shippable. Yeah. Yes, Eric.
Maybe the last question.
Okay.
On the Anthurium black nose.
Yes.
So just to ask a silly question, it seems like we have 3 different types of fungus that have the same symptom.
Well, it looks like right now we have 3 species of Colletotrichum and Phomopsis. Yeah. But we need to figure out really the true similarity or differences of the Colletotrichum. Because like I said, the molecular sometimes, sometimes it's, but they do look different. So I suspect they are 3 species.
So
and then...
has it always been here? Why are we getting a
Yeah, that's a
virulent
really
strain now?
good question.
Is it the climate, weather change? Because I think we're using new fungicide right now. We're getting results now.
Okay.
So that's a good sign. I mean, we have at least a Band-Aid here now.
Yeah.
We're losing like 50% of the crop once we get the black dose. So it's going down quite a bit now after about 4 weeks of spring. So I'm just kind of, is this just a temporary fix, or is it, I'm going to get reprieved a little bit, and then one of those 4 is going to get more virulent, and then.
Yeah, well, I think that's why it's important to know what's out there. I mean, and actually increase our population study to do those comparison of what is effective and not effective. But, on a bigger, I guess, the bigger picture, that seems to be very common in other crops we're studying. This, you think it's one thing, and actually then we find it's a different species mimicking what we're used to working with. So it's not just Anthuriums. Pathology, it's the host, it's the pathogen, and it's the environment. And that sounds simple, but it's actually very complicated. But that whole environment thing, I mean, it can change a degree. And actually, a fungus that never caused a disease now can cause a disease. So that's a lot of, that area to really get a better understanding. Yeah.
Change going on.
Yeah. Yeah. Okay.
For me as a commercial grower, just count our blessing. Now we got a fungicide that's working, and then.
Yeah, but I think just this continued working together, right? That, and especially getting the sample even into Brian. You want to make sure you are treating what you think you have, right? And then the information, getting the information back to you that, hey, these fungicides work well, and actually these don't. So when you know what your populations are, you know what to use at what time. Yeah, and hopefully we'll start to have better clues. Maybe we can tease apart the symptomology too. That right now, on first glance, it looks like the same thing, but maybe it's going to be subtly different. That growers can look at it and actually see, oh, okay, that is this and not this, and know what to apply.
I just hope that it's the collaboration between CPAR and PBARC.
Yeah.
That you guys are just, when you're working together, we're seeing more clear.
Yeah, definitely. It's like the line with me and Brian.
Yeah.
Tell him he can't.
That's why I'm saying,
Yeah.
Wait a second. Oh, you're right. You're right. Oh. Yes.
We don't want him to retire.
No. I guess last question.
Getting back to ROD.
Yes.
The spread of it, water, wind, beetles, or the board.
Okay, well, firewood, people, that can kind of be managed. The main way is infected boring dust of beetles, okay? That's the one you can't really control. When the infected trees are attacked by beetles and this boring dust comes out and there are fungal spores in it and they travel however far, that is what is more challenging. And that's really what directs management as well. In certain areas, there are little to no invasive beetles, so there's no activity. There's no generation or freeing of these spores. That's one case. In other cases, unfortunately, there's a lot of boring dust. So what do you do in that case? But that's really the challenge. We don't find it in the soil typically. I mean, unless you see a lot of boring dust in the ground and now you sample that soil, that's when we've been able to find it. But it's still good, for that and other things, weeds and pathogens. I mean, all the sanitation protocols that are suggested, that's just good practice and cuts down on a lot of things. But unfortunately, it's that wind-blown, but different than the rust wind-blown. Okay, the fungus itself is not wind-blown. That's why rust spreads so fast, so quickly. Their spores just kind of, you take the dandelion, they're gone. That's not Ceratocystis. It's attached to these boring dust particles or sawdust particles, or that material getting caught in soil in your boot, right? But again, you know, it's that philosophical question of, well, what's the calculated risk of that one thing, finding something new somewhere and actually starting something? I mean, a whole lot has to happen in the right way at the right time to have a new infection.
The borers controllable by pesticides?
The entomologists are looking into that. You know, that's really the idea of either tarping material once it's felled, getting those trees down on the ground if it's pumping out boring dust. I mean, to really keep it a very small spread instead of getting up into the wind. That's a lot of work in progress. Yeah.
Is there an upper elevation limit to beetles?
Oh, to the beetles, yes. So it really depends on where you are on island. I mean, it varies. I mean, there's native beetles and there's invasive beetles, and they all have their ecological ranges of where you can find them and where you can't. We really don't suspect that the natives are playing a role. They do things very differently, but it's the invasives, and then we tend to see, you know, when it's next to eucalyptus or next to the strawberry guava, you know, where there's this reservoir of all these beetles anyway, that's when it's, okay, now the ohia get attacked a whole lot more. So it's a challenge for sure. Okay. Thank you very much.